Hello, I'm Mike Ruggieri. Thank you for inviting me for this talk. This is my disclosure. No, so I've been tasked with explaining what we've been doing with bladder reinnervation of the past 20 years in the next 10 minutes, which is a bit of a daunting task. But I thought I'd just give you some of the high points. We started in 2000 with Randy Betts at the Shriners Hospital of Philadelphia, who thought that we could do a intercostal nerve transfer to reinnervate the bladder. And the first grant proved that it was not feasible to use an intercostal nerve. And so we started to use a caudal nerve that innervates a tail. And so the second round of funding from the Shriners Hospital was a different title, somatic nerve transfer. Um, and then there was a symposium by the Belmont Hospital, Ken Peters, in 2009 that I attended, and I met Justin Brown, a neurosurgeon who joined that project at that time. And then after that, Dr. Mary Barb was tired of volunteering her time for the Shriners funding, and we joined together to uh, do a joint applications at NIH, and that's been funded for 3 consecutive 5-year cycles. Okay, so this is the experimental design that we settled on after 20 years of experimentation. In order to show that you can reinnervate the bladder, you first have to demonstrate that you functionally denervate, or actually decentralize the bladder, because the bladder has nerves in the bladder wall cell body, so you can't really denervate the mammalian bladder, except rodents perhaps. Um, so what we decided to do initially was just cut everything below L7, um, and it turns out that that did not eliminate micturition behavior in these dogs. We have a video camera over each one of their housing cages. So we did did a group of 8 animals where we cut, in addition to all the sacral roots and everything below L7, as well as the hypogastric nerves on both sides, the L6 and L7 dorsal roots. Those animals did not show micturition or defecation behavior, but they did not do well, and they had to be euthanized early, so we could not really study them. So we settled on this method of we cut just the dorsal root of L7, as well as everything below L7 and the hypogastric nerves. And about 70% of those animals do not show micturition or defecation behavior, or they don't show it unless they have a urinary tract positive bacteriuria, which I'm not going to be able to discuss in great detail. So then this study that I'm going to talk about today, we have 14 controls. That includes 3 normal animals and 11 sham operated controls that were gone for a period of a year. Then we have 4 animals where long-term decentralization, where we just did this decentralization procedure. And then in 8 animals that did not show the behavior, in 9 to 13 months after the decentralization, we reinnervated the bladder with obturator to pelvic nerve transfer. Branches of the obturator nerve, about 25% of the fascicles, actually on both sides. We also reinnervated the pudendal nerve with branches of the sciatic nerve, which I'm not going to have time to talk about today. Okay, so I should say that we have already done this in a number of animals where we did the nerve transfer immediately after decentralization, or at 1 month or 3 months after decentralization. And we had pretty good results with reinnervation after that. So this is long-term decentralization before reinnervation as a preclinical trial before we do this in people, because if you have a patient that has a lower motor neuron bladder by some kind of traumatic or surgical event, you're not going to do the surgery to reinnervate the bladder until you know there's something wrong with it. So the longer period of time between the decentralization and the reinnervation, where it's possible, the better it is clinically, whether you can actually do this in people. And so that's why we focus in those most recent study on these long-term decentralization. And this is an intraoperative view of the dog bladder here, showing the obturator nerve as it passes real close to the pelvic plexus. And this is the transferred obturator nerve indicated at the end of these scissors. And so what we did is we stimulated the obturator nerve before the anastomosis. So the electrical stimulation had to go through the anastomosis to reach the part of the obturator nerve that was reinnervating the bladder. And this is the actual detrusor pressure that we measured. And this is if you actually stimulate in the unoperated animals the actual anterior vesical branch of the pelvic nerve, you get a very good bladder contraction, generating about 20 centimeters of water pressure. In the decentralized animals, when we stimulated that pelvic anterior vesical branch of the pelvic nerve, we got much lower contraction. Two of them we didn't get any contraction, and two of them we got a very low level of contraction. Unfortunately, we didn't get a lot of responses in the animals that were reinnervated 1 year with the obturator nerve transfer. We got a little bit higher in two of these animals than the sham operated controls. And it turns out that the actual detrusor pressure that we were able to measure in response to electrical stimulation, functional electrical stimulation of the transferred nerve, was inversely correlated with the percent of collagen in the detrusor layer. The inflammation score was the big driver of this inability to generate pressure. So the greater the inflammation score, the less bladder pressure we were able to generate. And it turns out that when we did the nerve stain with a protein gene product 9.5 stain, the number of ganglion per bladder wall were positively correlated with the actual degree of nerve stimulation induced maximum detrusor pressure. So at the end of this study, we also stimulated the entire spinal cord. We did a laminectomy all the way up the spinal cord to the T10 level and stimulated from individual spinal segments from L1 all the way down through the sacral region in both the sham operated, the decentralized, the 4 decentralized animals, and the 8 animals that had the obturated to pelvic nerve transfer 1 year after decentralization. And what we found is that, as you expected, if we cut all these roots, and so when we stimulated those spinal segments, the actual spinal cord in this case, we got no bladder contraction, which was perfectly what we expected to find in both the 4 long-term decentralized animals and in the 8 animals that were long-term decentralized and then re-innervated with the obturator nerve transfer. And then in the sham operating control, we got a robust contraction in these segments. So we combined the responses to L7 to S3 to just to simplify this data to show the maximum detrusor pressure generated by stimulating any of these segments. The upper lumbar segments from L1 to L6, we got, and this is the actual origin of the obturator nerve. The obturator nerve generally comes from L3 or 4 to L6, and it's highly variable from animal to animal. And so this is a source of the nerve that we transferred to the anterior vesical branch of the pelvic nerve in these animals. And as you can see, we got a fairly good contraction in a couple of these animals, but only really in 3 of them was the response much greater than the 4 long-term decentralized and 2 of the long-term decentralized animals. Even in the sham unoperated controls, we got a fairly decent contraction when stimulating these upper lumbar segments, and this is almost exclusively as a result of stimulating the L2 root in these sham unoperated controls. And remember, these animals did not have an intact hypogastric nerve, and we confirmed that with staining for tyrosine hydroxylate in the bladder wall, showing that the hypogastric nerve did not sprout and reinnervate the bladder during this time. So we don't think this is a result of stimulating the hypogastric. We think this is a direct innervation of the bladder by the upper lumbar region, primarily the L2 root. So as we try to clinically translate this, the first patient group that we planned on doing this is a patient that has a chordoma of the sacral cord, where the tumor completely surrounds the sacral roots. And in order to save the patient's life and remove the tumor, they have to also remove the sacral roots, which is very similar to our dogs. So this is a human cadaver showing that there is indeed a major branches of the pelvic plexus that go towards the. These are the anterior vesical branches of the pelvic nerve in a human cadaver. showing that this surgery is indeed possible in humans. And we're planning on doing it up in Boston with Justin Brown at the end of May.